Now let us have a look on how the disturbances in these genes leads to our respective syndrome. You should keep in mind that this syndrome can be transferred from either parent, the mother or the father, to the offspring that has the meiotic non-disjunction at the M1 phase. Now let's have a look at the underlying pathophysiology in males. In males at the normal meiosis 1, what is happening is, in the process of spermatogenesis, the XY chromosome, it splits into the cells having just an X and the cells having the Y on other hand. So we have these two varieties at this particular level. Then at the step of the meiosis 2, we have the formation of these two variants, the sperms having the X in them and the sperms having the Y inside them. Now, when these kind of sperms are going to get fertilized with the female oocytes carrying the X in them, after the cleavage, they're going to form the cells having a genetic makeup of X, Y, or it can be an embryo carrying an XX. So this is the normal mechanism that is happening. Let's take a look what goes wrong in people that present with Klinefelter syndrome. So here you can see that at the level of meiosis 1, the XY during the process of spermatogenesis is going to encounter a meiotic non-disjunction over here. So this is the core cause for the formation of Klinefelter syndrome. We have a cell which is empty, as you can see over here. And then we have a cell carrying the XY at the level of meiosis 2. So this is going to form sperms carrying an XY inside them. When such sperms are going to fertilize the oocytes carrying the X chromosome, they're going to form a genetic makeup of XXY. And on the other hand, it can also result in the formation of a genetic makeup of, after cleavage, of cells having just an X in them. Now, this XXY is responsible for the Klinefelter syndrome that we're discussing today. And on the other hand, this X, the people who are carrying only this X in them, they are going to present with a syndrome which is known as the Turner syndrome. So this is very important to note down over here and keep in mind these genetic makeups and how they're going to phenotypically present to you in your outpatient departments. So I hope it's clear now that the underlying pathology responsible in males is this meiotic non-disjunction that is present at the level of meiosis 1 over here. Similar mechanism is followed in females as the cell will transfer both of its XX chromosomes to a single cell and the other cell will remain empty. At the normal mechanism, in a female at the level of meiosis 1, there is a formation of an oocyte carrying one of the X chromosome, and on the other hand, we have a polar body which is carrying the other X chromosome. After fertilization with the sperm, the genetic makeup could either be an XX or either it could be an XY for the males. But what happens in patients who are presenting with Klinefelter syndrome at the maternal level, at the level of meiosis 1, there is the formation of the XX in the oocyte. Both the X chromosomes are being transferred to one of the oocyte, and the polar body remains empty over here. After fertilization with even a normal sperm, the genetic makeup in these patients would be an XXX or either an XXY. So this is the Klinefelter syndrome. If you could identify this as Klinefelter syndrome is present only in males, so this is the genetic makeup for the Klinefelter syndrome that we're talking about. This is another syndrome which is present in females and is identified by a genetic makeup of XXX. So I hope it's clear now that even on the maternal side, a meiotic non-disjunction is basically the underlying cause for the development of Klinefelter syndrome in these patients. Now, you might be wondering, why does a normal meiotic egg would show such changes that leads to this issue? The fact is there is no established reason for this statement, except that increase in the maternal age can be one of the risk factor which can play a major role in such meiotic changes. Even some of the studies carried out showed that when the maternal age exceeded 40 years, the patients presented with Klinefelter syndrome was also increasing. So the incidence was basically related to the maternal age.